Copper. It's the 29th element in the periodic table, and a common metal found in everyday items ranging from wires to cookware. It's also used by the body as a mineral required for multiple functions. This includes playing a crucial role in cellular respiration, the process that produces energy in our cells. However, our bodies only need a small amount of copper to survive, about 100 milligrams. Excess copper, mostly from our diet, is usually filtered by the liver into the urine and bile, which is a component of feces, from which it can be removed from the body. If the liver is unable to do this, then the copper will build up and can damage organs. This is what happens in Wilson's disease. It is a serious and potentially life-threatening genetic disorder affecting approximately 1 in every 30,000 to 50,000 people. In Wilson's disease, a genetic mutation causes the body to synthesize a defective form of a protein called ATP7B, which is primarily found in liver cells. Normally, this protein transports copper to different sites inside a cell, including to enzymes that require copper to function, and to the cell membrane in liver cells, where excess copper is then expelled from the cell into the urine or bile for excretion. The defective ATP7B protein in Wilson's disease results in impaired removal of copper and thus its accumulation, as liver cells are unable to transport copper to the cell membrane for excretion into bile or plasma. It also disrupts the function of copper-dependent enzymes in cells. Wilson's disease results from mutations in the Wilson's disease gene, ATP7B, which codes for the ATP7B protein. This gene is located on chromosome 13 in the cell nuclei of humans. In Wilson's disease sufferers, over 300 mutations in the gene have been identified. These are mostly substitution mutations, where a DNA mutation results in one amino acid, the building blocks of proteins, being substituted for another, which can drastically change the protein's functionality. You can think of it like changing the color on a Rubik's Cube tile. Just one seemingly minor change can make the entire cube unsolvable. In each cell, humans have 2 copies of each chromosome. Wilson's disease is a hereditary disease, meaning it is passed on from parents to their offspring. It has an autosomal recessive pattern of inheritance, meaning the ATP7B gene on both copies of chromosome 13 must be mutated for a person to have Wilson's disease. If a person only has a mutated gene on one copy of the chromosome, then they are a carrier, with the risk of passing the mutated gene to their children but otherwise are healthy, because the normal copy of the gene produces enough normal ATP7B protein to maintain normal function. Wilson's disease is potentially fatal and has widespread effects on the body. Copper primarily builds up in the liver and brain. Inflammation, severe scarring, and cell death occurs in the liver, which can cause abdominal pain or swelling. Damage to the liver impairs its ability to remove toxins from the body, causing fatigue and confusion. Jaundice occurs due to accumulation of a body waste called bilirubin, which is normally removed by the liver. This liver damage can progress to liver failure if not treated. About half of Wilson's disease patients suffer from nervous system disturbances. These include cognitive decline, memory loss, impulsivity, and impaired decision-making. Symptoms similar to Parkinson's disease can also arise, such as slow movement, lack of coordination, loss of muscle tone, and rigidity. A characteristic feature of Wilson's disease is the presence of Kaiser-Fleischer rings, KF rings, and sunflower cataracts in the eyes. KF rings are dark copper deposits that encircle the outer edges of the cornea, the colored part of the eye, where it meets the sclera, the white of the eye. Sunflower cataracts are cloudy green or brown discolorations of the lens. However, both of these features do not cause major disturbances of the vision. Wilson's disease can also cause weakness of the heart and infertility. Multiple tests are available to diagnose Wilson's disease. The gold standard test is a liver biopsy, where tissue from the liver is extracted and examined for degree of damage and extent of copper buildup. The levels of copper in the blood and urine can also be measured. Blood copper levels are found to be low in Wilson's disease, while urine copper levels are high. Moreover, levels of ceruloplasmin, the main copper-carrying protein in the blood that relies on ATP7B to uptake copper, can be analyzed and are often low in Wilson's disease. The eyes may also be checked for KF rings and sunflower cataracts. As Wilson's disease is hereditary, genetic testing of the ATP7B gene may be conducted to screen for associated mutations in individuals with a close relative who has Wilson's disease. Treatment of Wilson's disease is usually effective. It aims to reduce copper levels in the body and alleviate the symptoms. Patients may be prescribed low-copper diets that avoid foods such as chocolate, mushrooms, and liver. Medications are used such as penicillamine, which binds to copper and allows it to be removed via urine. When copper levels have restabilized, zinc is often taken, as it activates a protein which prevents the absorption and transport of copper into the liver by binding to it. In patients with liver failure that is not responding to conventional treatment, liver transplantation may also be used as a last resort.